coming, bright and early. I'm really pleased to welcome you all to the Center for Functional Medicine's Grand Rounds, and we have a wonderful guest today from the Institute for Systems Biology, Dr. Nathan Price, who I've known for a while, who's doing pioneering work in the area of systems biology. I'm going to read this because I can't remember it all, so much stuff. He's a professor and associate director of the Institute for Systems Biology in Seattle. He's also affiliate faculty in the departments of bioengineering, computer science, and engineering, and molecular and cellular biology. That's a mouthful. At the University of Washington. He's a co-founder and on the board of Aravel, which is a scientific wellness company. And he's also won many, many awards and has published over 100 peer-reviewed articles and serves on the editorial boards of many leading scientific journals, including Science Translational Medicine and Genomics, and Cell Systems. He's also served on the National Academy of Medicine's Committee to review omics-based testing to predict clinical outcomes in clinical trials. And he's also on the advisory boards of many, many companies and programs. So welcome, Dr. Price.
Thank you. Thank you, Mark, for that kind introduction. So today I'm going to tell you a little bit about what we've been doing out in Seattle at the Institute for Systems Biology. And it's about a topic called scientific wellness. And I'm going to give a little bit of a definition about that and give you a view of data and sort of how we're dealing with those issues. So objectives from this. One, we'll explain the basis of scientific wellness as a proactive approach for enhancing wellness and predicting and preventing disease. Two, I'll demonstrate the use of what we call personal dense dynamic data clouds to drive biological and medical discovery. And then third, I'll talk about describe a vision of healthcare that is predictive, preventive, participatory, personalized and participatory, what we call P4 medicine. Disclosures, I think we already went through that. I'm a co-founder of Aravel, and they helped to fund part of the Pioneer 100 project that I'll talk about a little today. All right, so this is Larry Smarr. Are people familiar with Larry here? Do people know who this is? A few people do. So this is a picture of Larry. It's taken from the cover of Technology Review from a few years ago. And Larry is kind of a prototype for the patient of the future. Depending on what kind of doctor you are, I guess you could be a dream patient or a nightmare patient, depending on what you like. But what Larry has done is that he started to measure all kinds of things about his body to an extreme degree. Might be the most measured person in the world. And what Larry has done was to essentially track himself over about the last 15 years. And several years ago, he had an interesting experience, which is a number of his markers jumped. They changed very dramatically from what he had seen before. If you go and visit UCSD, he has, I should mention, Larry is the head of the San Diego Supercomputing Center. So he has resources that are not usual for most people. And so you can go visit him, and he'll have a wall that's about 4 times the size of this room, where you can view all the data that he has on his body. So it's kind of impressive. But anyway, what he had at that time was a number of his markers changed. He delved into them. He went to his colleagues at UCSD, did some science, goes into his doctor, and he says, I think I'm getting inflammatory bowel disease. And his doctor said, how do you feel? He says, I feel fine. His answer, well, then there's nothing we can do for you. And he went to a number of different doctors, and he got sort of the same response. Now, that's not really a criticism of the doctors. That's just a way that our healthcare system is set up. For the most part, we respond to care. We do care after illness. We respond to symptoms. And they weren't really set up or ready to deal with just having data about a system saying, okay, we think that this has gone awry. And so what we've wanted to do at ISB is basically take this experience of Larry's, which was an anecdote, right? It could have been different. I haven't finished the story. So anyway, he got that response. So Larry went home. A few months later, he had an acute episode. He ended up having to go into the emergency room, got treated, got diagnosed, and he had IBD. So that's an anecdote. It's a story. It could have gone differently. It could have been, didn't have to be true. But in his case, it was. And so what this does is I think it's evocative of the kinds of signals that might exist in our body that are presage disease. And what we want to know is to what degree do those exist, to what degree are they actionable. And so what we wanted to do is to go from this story, this anecdote, this experience of Larry's, and expand it out to a large number of people so that we could really make this into a science and really try to study this. So this is what we've been doing. So we have, as I think everyone is aware, an aging population. And so this is not really a problem, right? Humanity worked really hard to achieve this. This was the point of clean drinking water, antibiotics, vaccines, all the work that's been done in science over the last centuries. And basically, though, we've achieved this. So we now have an aging population. The problem is that along with this, we've also seen a rise in chronic disease. And in fact, the CDC estimates that 86% of total healthcare costs are attributable to chronic disease. So we have a huge issue. And of course, places like this are really geared up to deal with some of those issues. So if we think about then the determinants of health in the United States, and this comes from a 2007 study out of the New England Journal of Medicine, they estimated that about 30% of a person's lifetime health is attributable back to the genetics, just the hand that they were dealt. About 60% of a person's lifetime health is attributable back to environment and lifestyle, and only about 10% of a person's lifetime health is attributable directly back to the healthcare system. So one of the things that comes to mind, of course, is that we need systems that focus more on the 90%. So if we think about healthcare and the way it's set up in the United States, I think we're very familiar with this number. We spend just under $4 trillion on healthcare in the United States. The National Academy of Medicine estimates that 97% of those dollars are spent on care after illness. So there's very little that's done for wellness or prevention in that sphere. There is, of course, at the same time, a wellness industry, and that wellness industry has, I'd say, a lot of good elements to it, and it has a lot of bad elements to it. There's a lot of aspects of the wellness industry that are not heavily supported by scientific data. And so what we see and what we believe now is that we're seeing the rise of a new industry that we call scientific wellness. And so what do we mean by that? So scientific wellness is essentially the idea of taking the same sort of wellness bent, which is that you try to optimize health, enhance health, rather than just waiting for symptoms and late-stage disease to come forward, but with a deeper scientific basis. And that deeper scientific basis, I would say, has become much more possible in the last years. This is being made possible by things like, I don't know how many people wear fitness trackers, omics data, the rise of systems biology, the rise of the ability to do genomics, proteomics, metabolomics, the ability to track electronically much more about what's happening in systems throughout the body and even in early stage healthcare. And so all of that together, we argue, is forming a discipline that we call scientific wellness. And that's been what we're really focused on. So this relates obviously to medicine. And in fact, one of the things that we'll argue is that scientific wellness and a focus on scientific wellness will really be the key to unlocking disease in many ways as well. Because if you have these kind of data and you're generating them over time as a function of trying to optimize wellness, it gives you a window or a mirror, a window into understanding transitions to disease. So you end up getting early data. I'm going to talk more about that. So this relates then to a concept of medicine, and this is the term that Lee Hood has coined. Lee Hood's the president of ISB. If you don't know who he is, Lee was the inventor of the automated DNA sequencer that made the Human Genome Project possible. He's the father of systems biology. He's the father of P4 medicine. Really accomplished person. One of only 15 people in the world who's elected to all 3 national academies. Won the National Medal of Science from President Obama a few years ago for this and so forth. Anyway, so Lee really coined this term of P4 medicine, which again is a medicine that is predictive, right? So we want to say something about what's going to happen. Preventive, right? It doesn't help you to know something's coming unless you can do something about it. Personalized to the individual, and participatory. And participatory really means empowering individuals who, so that they have the information they need to be the centerpiece of their own health. And what we see here is the rise then of a scientific wellness industry that focuses on one aspect, which is the quantification of wellness. And then the other side, which is studying disease, is really represented by the current healthcare industry, which is, of course, really a disease care industry. So one of the things that I'll argue is that when we study health and disease as researchers, we often don't study what we most want to know. And I'll use myself as an example. This isn't meant to point fingers at anyone. I'll point them right at myself. So we have a grant from the National Institutes of Health to study Alzheimer's disease. And we're learning a lot of great things out of that. But we mostly study the brains of people who have Alzheimer's versus those who don't. When do we do that? After they're dead. Right? No one's going to give you a piece of their brain before then. And so when you, what we really want to know is 20 years earlier, what was happening in the body of the person that got Alzheimer's that was different than the person that did not. And what we'd really, really like to know is if we had known about those things, could we have intervened? Could we have stopped that from going forward? And so in thinking about those kind of things, Lee and I, in 2014, announced or proposed a project that we called the 100K Wellness Project. And this was an ambitious goal. The idea is to collect 100,000 individuals, get whole genome sequence data on everybody, and then multiple times per year do proteomes, metabolomes, microbiomes, clinical chemistries, wearable devices, clinical labs, and so forth in order to generate data that is dense enough in terms of the number of measures that we're making, and then long enough and dynamic enough so that we can watch evolution of a number of conditions over time. And the goal here was to identify the early warning signs for all the major human diseases so we could develop ways to predict them and prevent them. Right? So it was a big longitudinal study. I will say that, and if you're doing the math in the back of your head, as I did when we were first talking about this, you're probably calculating to yourself, that's about a $10 billion project. And so when we announced this, we had raised $0. So I thought that was fairly audacious to say we were going to do this. And I remember having this conversation with Lee as we do this. And I said, because he had asked me, what kind of numbers would we need for statistical significance? And I remember sitting in my, I'm thinking, 1 million is crazy to propose, 10,000 is clearly too few, and I was like, 100,000. He's like, all right, we'll call it the 100K, the 100,000 person wellness project. And I argued with him a little bit because I'm like, Lee, if we say it's the 100,000, let's say we got 20,000 people, 50, like that'd be the biggest study by a mile ever done with these kind of data. And yeah, he kind of was a little frustrated. Nathan, it's 100,000. I was like, okay, so we announced it. It's the 100,000 person wellness project. And so what's been really amazing about working with Lee is kind of the power of determined optimism. And if anyone who knows Lee, those two words are probably the two that are most used to describe him, determined optimism. And with his history of success, that's proved well time and again. But what happened here then was that we were really able to launch this, and this has now become a real thing, and we actually think that this will be much bigger than we had initially visioned and taken on sort of in a different way. So the way that this is evolving is we've essentially implemented scientific wellness in two different directions. So one is that Lee and I, along with Clayton Lewis, who's the CEO, have founded a company called Aravale. And I won't say too much about them today, but just a little bit, because this is a kind of a vehicle of how this arose. So Aravale is a scientific wellness company. It is providing data of the types that I just listed, so these massively multi-omic data, and providing a program for individuals to optimize their wellness. And I'll talk a little bit about the founding project for that in just a moment. And so that's a company. We've spun it out about 2 years ago. It has about 180 employees now. It's raised just under $55 million, and it's serving now thousands of people that are coming through. One of the things that is happening there is that when people come through Aravale, they're given a choice. And if they check a box saying that their data can be used anonymously for research in order to help to learn about health for themselves, their kids, and their grandkids, then that data is anonymized and brought back into ISB where we can study it and try to learn from it. And so far, 95% of people have said yes to that. And we're very grateful for that because that really is the substrate of being able to identify so much that we want to understand about health that we don't know now. The second way that this is moving forward is that ISB, and this was very fortuitous for us when this came about, but ISB is now an affiliate of the Providence St. Joseph Health system. And so And Lee Hood has now become the chief science officer for that whole operation. And so if you're not familiar with this, Providence St. Joseph is one of the largest nonprofit healthcare systems in the country. It now has 50 hospitals, 13 million patients a year that come through. And so it is a major system. It's in 7 states. And so what this does now is that we're actually able to do a series of clinical trials, and I'll talk about a few of those within the Providence system, generating the same kinds of data. We're also able to leverage just an ability to interface with this large healthcare system. And so we have then people who are coming into these types of programs in very different ways or through research studies, both via the people that sign up for Aravail and check the box and say that we can use their data anonymously for research purposes, and as well as the individuals that come in via Providence. And so this is how this is all moving forward now in really a beautiful way. And so the beginning of all this was a study that we call the Pioneer 100. And so the Pioneer 100 was, when we were envisioning, that 100,000 person project, that's a lot to bite off. And so what we did instead, to start, was to get 108 individuals. It's 108 because people kept sort of asking to come on, and so we kept letting a few more people. One of them is a very well-known person here. So I've heard this is public here, but it seems to know, but so Toby Cosgrove was one of the people who's come out and has said that publicly. We don't tell the names of anyone in the study unless they themselves have come out publicly. So anyway, though, but we've had a number of the individuals who have come through, and so we had pioneers that have come through this, as we call them, come through the study. And so basically, then we collected a lot of data. So this is what we collected. So we did whole genome sequencing on everybody. That gives you millions of differences just at the SNP level between individuals. We did a lot of clinical labs, about 150 different clinical lab tests. These are really important because the clinical labs are what we more understand how to interpret, and so they really act as a Rosetta Stone in some ways of trying to understand the meaning of some of the other kinds of data that we gather. Oh, shoot. There we go. We did gut microbiome. Oh, and by the way, those clinical labs, those were 3 times a year. So this was a 9-month study, so we did it every 3 months. Microbiome, same thing. We did these out of gut microbiome 3 times a year. And then self-tracking, so this is Fitbit and things like that. So all these data together then are put into a database. And what we're trying to do in the short run is to identify actionable possibilities. What can we learn from the data that people can act on to make an impact on their health? And then on the long run, we're trying to make new discoveries about how these data are interrelated. And I'll talk about that more in a minute. And these are what we call our dense dynamic personal data clouds. Okay. So in the study, we also had coaching, and coaching is a big part of all of this so that there's a way to interpret this. We don't want to flood people with tons of data. We want to take that data, go from complexity, do analysis so we can get down to informed simplicity. And when we do that, then we want to make that turn into actionable possibilities for the individuals. And so our wellness coach was Sandy Kaplan, who was just fantastic. She now leads coaching at Aravail. That was part of the model that we did, this Pioneer 100 project. We recruited a lot of people in to help us do the study, and then many of them actually became the founding group over at Aravail. And then we had a study physician, Craig Keebler. So this was very much not a clinical study. It wasn't, this wasn't to provide medical advice to individuals, but we did have physician oversight. So anything that became medical was referred then back into the healthcare system. And so that's how this was set up. So just a couple of notes on outcomes here. So over the course of the 9 months, we tracked, as we said, a number of different markers and so forth, and we saw improvement across multiple dimensions for these individuals. So you can see here cardiovascular, those markers moved fairly slowly, so there was an improvement by 6%. This chart, by the way, is just the number of people who were out of range in round 1, round 2, and round 3. And so it just shows sort of a difference at that level. And so here, let's see if that works. And so it sets at 100%, just however many were out of range at the first level, and then after that you see the improvement. So we saw an improvement of about 6% on the cardiovascular markers. Diabetes improved by 37%. inflammation by 12%, and nutrition by 21%. So this is just based on standard clinical ranges. So we haven't redefined any ranges here. This is just based on normal clinical labs, and they're already given values. We actually do think over time that the values of those and how they're interpreted should be very personalized. And in fact, there's a lot of data now that shows that based on your genome, you can predict different levels of analytes for individuals. What is not yet really understood is if those differences in predicted levels mean that your target level is different. That's one of the evaluations that really needs to be done. And we actually think over the next 5 to 10 years that there'll be a remaking of blood interpretation. I think that basically in the future you won't want to interpret blood data without the genome. Because there'll be so much personalization that will happen there, but that's not proven yet. Those are all studies that need to go. But what is proven is that you can predict in a number of cases the level of certain analytes from the genome with some specificity, and it varies from analyte to analyte and so forth. But we're at such a nascent stage. We're just starting to get genomes on, thousands of people. We have not yet done, millions or billions. And we also haven't had these kind of dense dynamic data associated with genomes before, at least not all of them at once. And so there's a ton of different elements that we can do to bring that together. So these are standard measures. That's not where we're going to end. That's just where we're starting. But just to give a sense over the course of the study. A second element, and this will probably be familiar to an audience like this, but we also in the course of just the 9 months were able to observe even a few disease states. And so one of these was we had an individual, and this person was having arthritic-like symptoms in their knees and ankles. And this was a problem for them because they really loved to go hiking with their family, and they couldn't do that anymore. And they had concierge medicine actually on both coasts, given the nature of their job. And they had been given, I think, some drug for arthritis, but basically this wasn't going away for them. So they came into our study. We did this 360-degree view, and something pretty simple popped out. So in this case, when we looked at them, the ferritin levels in their blood were really high. So they had high ferritin levels, and then we go into their genome, and sure enough, they're homozygous for 2 genes that encode for the most elevated risk for hemochromatosis. Now, us not being physicians, I'd never even heard of hemochromatosis before, so the data just kind of pointed us there. We kind of learned about that. But we found that, and one of the interesting things, so hemochromatosis kind of mainline leads to liver dysfunction, and ultimately you can die from hemochromatosis. But a lesser known side effect apparently is that high iron in the blood can actually catalyze cartilage breakdown. And so some people get these arthritic-like issues. And so we referred him back into the healthcare system. He was in fact then diagnosed with hemochromatosis and given the treatment for that. Now, people in this room will probably know that when I give to science groups, they never know the answer. But the treatment to hemochromatosis is dead simple. You donate blood once a month. So he starts donating blood once a month. The iron levels in his blood, and I'll show that here, went from the red to the green, so they totally normalized. And the arthritic-like symptoms that he had in his joints went away, and he's back functional and with his family. And so it's an N of 1, right? You can't prove that was the reason, but that's what happened. So in essence, what you see here then is data. So this shows in the green, you see individuals that have 0 copies of the gene that gives the risk for hemochromatosis, and they're in the normal, and that's just an average. And the orange, you see if people have 1 copy of the gene, you see that's elevated. And in the red, you see the really high elevated levels for individuals, especially for this individual who had the 2 copies. Now, N there is small, right? So we're not learning this. We're just applying something that's already known here. But what's fascinating is that for the 2 individuals, the one is the man that I just spoke about. The other is a woman. And what's really fascinating there, so she's premenopause. So she's premenopause, so this wouldn't have manifested. And there's no guarantee. Genetics are not fate. It may never manifest. But what's happened for her, though, is that she now has just a heads up, says, okay, you're at risk for hemochromatosis. No big deal. Just monitor your ferritin levels. If they ever start going up, just make sure you donate blood, and you've essentially eliminated a disease trajectory from her future. And if you think about this case, in both of their cases, I think what's fascinating is that, in essence, because they have knowledge, they're empowered to make a very simple decision that removes a disease trajectory from their future. Okay? And we would like to do that for as many diseases as possible. So this ends up being a fairly simple example of that. All right, so now I want to switch modes here into data analysis. So we gathered all these data. Again, this is the first cohort for which all these data had been measured at the same time from the same people, same samples, and so forth. And so basically, what we're able to do here is take metabolites, clinical labs, proteomics, microbiome, lifestyle data from Fitbit and things like that. And essentially, what every blue line in this plot is it is a correlation that crosses across those data types that is statistically significant after multiple hypothesis correction. So this is about 3,500 correlations that go across this. So you can just start to get just a glimmer of how interconnected all of these different systems are. And so this is just the tip of a very big iceberg. So one of the next things we can do with that is that we can take that correlation network, and we've run an algorithm, and you'll see it just computing there right now. But basically, we run an algorithm that identifies modules out of that network that are very highly interconnected. This is done, I won't get into the details of the algorithm, but essentially, if you imagine a place, well, I'll use my hometown of Seattle, and if you have Seattle, there's a whole bunch of different neighborhoods, and they are connected by bridges. And if you run an algorithm, which is what this does, basically that's the shortest path from every house to every other house, it would identify the places that are traversed the most. Those are the bridges. And so in this case, what this algorithm does is it then progressively disconnects the bridges and then leaves you with the neighborhoods. And so this gives you all the neighborhoods. And so what this does then is it provides a contextualization for all the usual clinical markers that we have with all of the things that surround them that are associated with them across the different data types. And I'll just give one example. So here's just one. So this is the total cholesterol community. And so you can see on the right-hand side there just a number of different molecules that are the most associated with total cholesterol and that come into this community. I'm just going to talk about one of them. In one case here, cholesterol is negatively associated with thyroxine, and that's the first link at the top. Now what's interesting about that is that thyroxine is actually a drug known as Synthroid. And one of the side effects of Synthroid is that it has this effect of lowering cholesterol. And so what's fascinating here is that the data clouds essentially pull that out, and it's a drug class that is based on a compound that's actually endogenous in your body. So what we're investigating now, because we have many, many of these, is there are a number of compounds then that would be potential drug candidates for targets of interest. So when you find what is in these neighborhoods, some of them will just be associative, but some of them will actually have this sort of causal relationship like what we see with Synthroid and cholesterol. And there are different ways you can try to tease that out by looking at time variation to some degree, which is somewhat helpful, and mechanism, and then doing a study where you can really get at that causal level. But what this gives us then is a very interesting path into potential drugs, and these would be drugs that are safe within certain levels because you already know that they are in the body. So these are endogenous compounds. It doesn't mean that they're safe at every concentration level, of course, but essentially what you're trying to do is to understand levers that you have to push and pull on in order to change certain clinical marks of interest. And so we think this is a very interesting path to drug discovery. Here's a number of other interactions, different modules, and I'm just going to talk about one of them. So I'm just going to give you one simple view of this. So there at the bottom, you'll see a label that says inflammatory bowel disease. Now that label, inflammatory bowel disease, does not actually mean that we have people who have inflammatory bowel disease. But what that label refers to is the genetic risk for inflammatory bowel disease. So earlier, when I was talking about some of the notions behind proposing the 100K Wellness Project, there was envisioned, and certainly in my mind was a little bit of this notion that we needed this big number because we had to have a subset of individuals who would unfortunately develop, the different diseases, but so we could go back and try to figure out ways to do prevention. But it was kind of like thinking about one of those, I don't know what they're called, the little things that plink down. You're kind of imagining, the different subsets. But we had a bit of a paradigm shift as we went along that made us realize we could get a lot more from smaller amounts of data. And what that was is that because there's been so much work done in the community for GWAS analysis, we have genetic risk profiles now for about 60 to 70 diseases that are high quality. And if you look at that, what we've done is you can take that genetic risk and actually use it as an outcome variable. And so by doing that, what we do then is take everything in the dense dynamic data clouds and figure out what is associated with those different risks. And we call this the manifestation of genetic risk in the body. Okay, and I'm going to talk more about that. So the notion here is that a genetic variant in a test tube doesn't mean anything, right? It means something in the context of your body. And so when you have genes that are imputing higher risk, they must do something. So they must have, and what they do must have some reflection in other systems. They've got to be reflected somehow in either the metabolites or the proteins or the microbiome or the RNAs or something. And so what we want to do is when we take these very dense dynamic data clouds, the idea is then to identify what is related to that genetic risk. And what that does for you is it means that the study of every single person's data is relevant to the study of every single disease. Right? Not everybody gets a disease, thankfully, but every single person has a genetic risk score. And so you can now leverage all of that work that's been done in genomics and use that as a means for understanding these other data. I'll give one example. So one example here is you'll see there's a connection there between inflammatory bowel disease and cystine. Okay? So I'll just show that. So here's the correlation that we find. And you'll see here it has a row which is a non-parametric correlation of 0.44. P-value there 2 times 10 to the minus 6. And so that's a false discovery rate of 0.03. So very statistically significant. I didn't put it in this talk in the interest of time, but if you look at similar papers that have been published for these kind of associations in Nature Genetics and other places, this is about how the correlations come out. And so when you get this then, this is a association that says the higher genetic risk somebody is for IBD, on average, the lower the amount of cystine that person has. And remember, this is in a population. This is not a disease population. It's a well population. And these are people that range in age all the way from the 20s to 90s. Okay? So when we observe this, we then go back into the literature. And there's been some related studies. So in case control studies of people who have the disease IBD versus normal, and this is what's shown on the left, it turned out that plasma cystine came out as one of the top candidate biomarkers. It was very different in the case control population. A related fact that's shown on the right is for ulcerative colitis, which is a form of IBD. There was a relationship that's the lower amount of cysteine, cysteine is the dimer form of cysteine, was related to having a worse outcome. So the worse your IBD was, sort of the lower that was, the cysteine. And so this was fascinating. And again, remember that we find a very similar relationship, but in a set of individuals that are pre-disease. And I'm going to come back to that in a minute. The other element that I just want to point out to you is that we can think about a potential mechanism. So cystine sits upstream of glutathione when you map this into metabolism. And so what happens is that if you have, and there's actually another paper that shows us, if you have suppressed cystine, this has a negative effect on your glutathione pathway, which is what combats oxidative stress in the body. Oxidative stress is a trigger for IBD. So a hypothesis comes out pretty clearly, which is that over the course of your life, you have these genetics, you have this suppressed cystine, and that suppressed cystine means that you're less well able to deal with oxidative stress. So it's more likely that some oxidative insult that you have in your body will go outside of your sort of, your resilient zone or whatever you want to call that, and you're not able to deal with that, which gives you more likelihood to trigger to IBD. Now, if that's true, then you can monitor that. Then basically over the decades of your life, that's an explanation for why individuals with this particular genetic risk, how it manifests, and even more importantly, if that's really true, then you could imagine taking someone who is at high genetic risk for IBD, intervening with cystine or maybe some slightly more complicated version of how we'll make that pathway operate more better over the course of their life, and you can take someone from being at high genetic risk to being at low actual risk for the disease. And what we want to do is just to map this out for every single disease for which we have good understanding of the genetics, and so develop paths to prevention. Okay? So that's just one example. So let me just, so this manifestation of genetic risk, just a few takeaways here then. So one, we can separate then differences based on genetic risk, pre-disease, from disease onset, post-disease. Okay, and so remember, that study that was done, that came out with cystine as being a potential candidate biomarker for IBD versus not. With our data, we would actually cast doubt on that because we would say, well, it appears that this was all pre-disease. In other words, so it didn't have anything to do with the onset. It was probably there for a long time. But without those pre-data, you can't do that. So this is a way then to control, to essentially provide a correction factor for any case control study. And so just imagine this. So basically all these studies are done the same way, us and others. Right? You get a control group, you get your cases, and then when you have your cases and your controls, in essence, you recruit them and you match them based on age and sex and smoking status and whatever, and basically, and you get a group of people that get the disease from those who don't. But it stands to reason that the group that got the disease first was probably at higher genetic risk. And what this does is this gives you a mapping for all the differences that you expect as a function of the genetic risk that would be in that population independent of the onset of the disease. So you can actually start separating those out. As I already mentioned then, this also gives us a path for potentially finding ways to do prevention. So if we can understand the manifestation of genetic risk, then that gives us an ability to intervene in genetic risk. And that's really key. Oops. Okay, and then I already mentioned this. So that gives us the potential then to take someone who's at high genetic risk for a disease and maybe make them at low risk for the disease. And that's really where we want to go. Okay, so everything that I just told you about came out from the Pioneer 100 study. Oh, I should mention the Pioneer 100 study was published yesterday in Nature Biotechnology. So after a long slog, but basically we got that out. And so if you want to read all about that, that's out in the public domain. So now I'm going to transition to tell you a little bit about some of the things that we're doing now that we've moved from the 100 up to the 1,000 people or so that we had in the first year that came through the Aravail program. I'm just going to give just a little bit about this. So one thing that happens, and it's unfortunate, but individuals that go through any program, will get disease. It's funny, when I gave a very early talk to this at kind of a nice rest home in Seattle, and early on, and I gave this talk, and the first question someone raised their hand, they're like, "What are we going to do in society when no one dies?" And I'm like, you definitely misunderstood this talk. So people are going to get sick. We don't want that, but that is going to happen. So we quickly put that to rest. Okay. So anyway, as a preamble, so this is a person then that went through our program that unfortunately did get stage 4 pancreatic cancer. This is so they got stage 4 pancreatic cancer. Bert Vogelstein is the most cited cancer actually researcher in the world. Has done studies and estimates that at the time of diagnosis that someone has probably had pancreatic cancer for 5 years. So we believe that she came into our study already having pancreatic cancer, unbeknownst to her or anyone. So she was diagnosed in January of this year, and basically what we did then, though, is that we had 4 prior blood draws and 2 microbiomes. And so what we wanted to do is just go back and see could we learn something? Is there something there that we could identify? And so in this case, what was interesting is we found one particular protein, and you'll see it there. So what is shown there is the measure of this protein for every single individual that's gone through the Arabelle program, at least after the first year, because this is the first year data. And so you see that histogram. And what you'll see there is there are 4 extreme outliers. And those 4 extreme outliers are all measures of this woman that has pancreatic cancer. So, it's too early to be definitive by far, but the interesting thing is that what we observe is that this is a protein. It's actually made in the pancreas. It's in the Notch signaling pathway. It's related to cancer. And so it's a candidate. So this is a candidate it's a candidate biomarker. You'd have to see this a lot more before you would say that. But what's really fascinating is that with these dense dynamic data clouds, we do have an ability to go back in every single case and identify the outliers, and we're just starting to catalyze all of these. And we're actually developing some new approaches around this that we'll release in the next year or so. But basically, there's a ton that you can learn by mining what we call the extremes of these dense dynamic data clouds. And so you start to pull these together, and we'll be able to monitor multiple pathways in to this disease. So that's just one brief example. We have a number of these now. And in fact, even in the well population that's in Arabelle, and this is from the first about 1,200 people or so from the first year, then there are about 70 different diseases that are represented. Some are small and some are larger. But there's a database to analyze there. Okay. So another thing that I just want to talk to you about a little bit is here, this relates to looking at metabolites. Okay, so these are metabolites. We measure about 700 of them. And what we've done is with collaborators is to develop approaches to look at metabolites that are out of range in different ways. Okay? And there is no pre-specified range. So this is just outliers essentially against a distribution. So when we identify these outliers, one of the things we did was we defined a healthy population. In this particular case, we defined it by people that reported to be disease-free, younger people who are reported to be disease-free in this case. And then what we wanted to do was just to say, okay, so we built these background distributions and then just asked, all right, how many different metabolites is any particular individual a outlier in? And then when we did that, we just started counting that up by decade. And so this we thought was pretty interesting. So if you look at this, this is just a box and whiskers plot, and it's going through the decades of life. So it looks at people in their 20s that have a low number here, onto their 30s, 40s, 50s, 60s, 70s, and 80s. And what we see is that on average, there's this monotonic upticking of the number of values that become divergent or that are outliers. And so this, I think, is pretty fascinating because it suggests that there are these kind of dysregulations that happen as you get older. Now, not all of these are dysregulations. Some of them are, in fact, things that need to happen as you age. And so that's one of the things we're trying to sort out. And there's some really interesting examples on that front as well. So that's what we see. So another thing that we did, and this is maybe getting a little more technical, but I just want to give you one big point out of it. So each one of these little sets here that you see is just like, this is a plot by decade of just what you saw on the last slide. But now what it's doing is that we're redefining what it means to be an outlier. So in the far left bottom corner, that's, outlier has to be, just at the very, very extremes. And then we're just widening it, and we widen it till the end, till we're actually saying that an outlier, and it's definitely not an outlier, but we're saying that this divergent value is anything that's in the upper or bottom 20%, right? And what's really fascinating is that relationship with age holds absolutely solidly no matter how far we go. And so what that suggests is that there may be very subtle kinds of regulation, subtle differences, that actually make a difference in the long run. So this is one of the things that we're trying to get into and prove because it's really fascinating that just the number of those changes really increases, and it doesn't matter at what level of cutoff you say when something is extreme. It makes absolutely no difference in the data we've looked at. That pattern is extremely, extremely strong. So we're just starting to do this and look at that. But I just wanted to give you just a little bit of a sense for where some of the data is going. And there's a mountain more that I'm not talking about right now. Okay. So in the last minutes that we've got, I just wanted to take you through a little bit of where we're heading and what we're looking to do. So with the Institute for Systems Biology and our new affiliation with Providence, St. Joseph Health Care System, we've initiated a series of what we call translational pillar projects. And these are projects to try to take ISB science and translate it into the clinic. So one of these areas is, of course, scientific wellness, which is what I've been talking about today. And so with scientific wellness, there's two big initiatives that we're pursuing. One is that the Providence system has given an opportunity for 1,000 of its employees to go through the AirAvail program. And so for the 1,000 employees that are doing that, what that is going to do is we'll basically take that and then compare it against a control group. And we're going to be looking at health outcomes for individuals for going through kind of a scientific wellness program. And we're also going to do analyses of economic impact. And what we're hoping to show is that there's an economic benefit to being really proactive that is offset by downstream disease avoidance. And so those are two things that are ongoing, just started now. The other element under scientific wellness is that Washington State University has started a new medical school. And that medical school is actually offering, again, the AirAvail program to all of their incoming medical students. And so Washington State University will take all of their incoming medical students. They'll have a chance to go through this program. And Lee Hood and I and a few other people are just about done now with a textbook on systems biology and systems medicine that also provides a foundation for some of the concepts that we've been talking about. And then they're looking at weaving then this personal data that all these doctors in training are getting right into their educational program. So they have a glimpse of what's coming in the future in terms of genomics, what it can do, what it can't do. Same with all the dynamic data, so they can have a sense of how to interpret these data, what are the limitations, what are the promise, what's the potential, and it puts them right at the leading edge of understanding that. And we're definitely looking for other groups that would like to be involved in that. We're also pursuing a clinical trial on Alzheimer's disease. And in fact, that there's at least some chance we'll do it with the folks here as well. But basically, for Alzheimer's disease, we are doing two different studies on this. One is related to work that Dale Bredesen has done. I believe he was a speaker here last year or so. Dale has a really innovative, very highly personalized program that has shown great efficacy in a series of case studies that Dale has done to date, but there hasn't yet been a prospective clinical trial. So Providence has given us $10 million in order to get that going, and so we're going to do a prospective trial of a program that is highly personalized, very integrative, and tries to take a deep dive into multiple different kinds of Alzheimer's. We're also going to do a trial with the Hoag Hospital and Rod Schenkel and the Hoag Hospital, which is part of the Providence Network, which will really be a testing of scientific wellness in a different population as well, and to see if that has an impact on cognitive decline. So that's an area that we're very interested in. Another one that we're launching is around breast cancer survivorship. And so here, one of the things that we're very interested in is, so the scientific wellness paradigms has worked well for a number of people just coming out of the general population. But one of the populations that is really at great need and has been brought up actually in, by people in audiences and talks and things like that before as well, is that when people go through cancer, even if they survive, they've gone through chemotherapy or radiation, and those all have decimating effects on the body, right? You'll take them because you're trying to get rid of the cancer. But what we'd like to do is to have a program that helps people get back to health faster. And so this is going to be done with the Swedish Cancer Institute in Seattle. And basically, we will take a group of patients that once they have finished, in this case, chemotherapy, we will then generate all the dense dynamic data clouds, try to provide coaching and working with the physician team in order to get them back to health, and also just to monitor what are all the different things in their body that have been decimated by going through that program and try to get them ways back to health. So that's another major initiative that we're looking at. And what unifies really all of these programs, and there are more that I have not talked about today, is this focus around these personal dense dynamic data clouds. And the data clouds are, we think, extremely useful to study wellness, but also to do clinical trials. And in fact, one of the things that we're arguing around to pharma companies and so forth, is that you really should just do these on every clinical trial. And the reason I say that is if you're investing, you invested your $1 billion into making some drug, what you're really going to want is to have these kind of data so that if you have a failure on your primary outcome, you have some ability to go in and figure out why that was, or what that was, or even if you have a success on your primary outcome, maybe there's a subset of people that really have a highly adverse effect, and that's going to block it. You'd want to know why that is, and having all these dense dynamic data gives you a window into that. There's also huge synergy because if you do these sorts of data clouds and you do them uniformly across all your trials, every single disease and every single drug, every single one of those things is a perturbation to the human system. And if you're generating these kind of uniform data and you have them in an integrated system, you can do learning across that human system in every case. And so the power of your clinical trial when you have these kind of data is not just from what you learn from the 200 people that are in that trial, but it actually could be learned across the tens of thousands of people for whom you have done these across different kinds of trials. So we think that this would be transformative to the whole process of drug discovery, the process of biomarker discovery, et cetera, et cetera. And so although there's a cost associated with these measures, we really strongly believe that if you deploy them and you have an integrated system where you can harness the value of all those things together, that it's really, really worth doing. And so this is one of the things that we've operationalized now. We have the software in place. We have the data, the ability to generate the data. And so this is really, I think, where the field needs to go. Okay, and these personalized data clouds, of course, will be the foundations of precision medicine, P4 medicine, scientific wellness. Okay, so in terms of a lifetime journey then, we certainly think about this, and Lee Hood likes to talk about this one a lot, but kind of where you reside on this wellness staircase. And the idea here, of course, is that scientific wellness is a lifetime journey, something that we can be involved in and try to optimize health no matter where we're at, no matter what age we're at. And the argument here is that the average person is maybe is not as high on that scale as they could be, but by finding actionable possibilities, you can continue on upwards. And what you'd really like to do is to become one of what Eric Topol calls the welderly, which are a set of individuals that have been haven't never been hospitalized in their life, have been basically healthy all the way through until very late age. There's a number of these kind of people, and so the notion is how do we get more and more of us to be a part of the welderly. So in terms of the future of healthcare, so this is where we're going. So we really want to bring P4 medicine to the healthcare system. The new affiliation with Providence is a huge part of that. Precision medicine, this is really employing these personal dense dynamic data clouds to each individual, and it really lets us understand both the genetics and the environment and how those things are coming together to drive health. We want to optimize wellness through actionable possibilities. We want to really be able to reverse disease at its earliest point. We see this as the preventive medicine of the 21st century. And these kind of dense dynamic data clouds are going to give us the substrates for thinking about how to do that. And we're just at the very tip of that iceberg. Scientific wellness, already mentioned, really should be a lifetime journey. We really hope that this kind of an approach, although there are the upfront measurement costs, we hope that in the aggregate this will really lead to a dramatic reduction in healthcare costs by reducing some of the really high acute care costs that we see. And so that is our hope, that this will be dramatically reducing. And also the amount of learning that is going to be done off this is just going to give us insights into the system in powerful ways. And we'll actually learn what's useful to measure and what's not useful to measure as we do that. We do believe that this is going to create a scientific wellness industry. We believe that that industry ultimately will be larger than the healthcare industry. So we do think that healthcare is going to sort of shift over time to be in more of that kind of a mode. And that there'll be a major transformation, and this is already starting to happen, but just with new technologies and focus, there will be a major transformation of the biotech industry, the healthcare industry, and so forth that's really coming. All right, with that, let me thank all the wonderful people that have been involved in this work. Obviously, Lee Hood, who's been an amazing mentor and colleague and partner through all of this. Fantastic people all throughout. We could never have done this project without bringing in a lot of people outside our usual scientific circles. Jennifer Lovejoy really designed the clinical-facing part of this and did a fantastic job. Sandy Kaplan, I already mentioned, and Greg Keebler. Sean Bell, not a scientist, but our business director, really helped put a lot of this together and really deserves a lot of credit for that. Analytics, I have to give huge thanks to Andrew Magis and John Earls. They really did basically every computation that you saw. For the work on the Nature Biotech paper, Andrew really should be sainted or something. He was so much work for years. Really did that. Anyway, project management, communications, our great medical advisory board. And I'll also thank a couple other members of my lab that were involved here, Roe Levy, Andrew and John I already thanked. And with that, I'm happy to take questions, and next grand rounds is coming up. Thanks.
Thank you. That was great. Thank you for your time and insight and amazing work. It's just interesting to see what the future of medicine is going to look like. So do we have any questions in a few minutes? Yeah. Dr. Calabrese.
Fabulous presentation. So, this notion of data cloud influencing medical decision making. I know you're thinking about this. So, you take immune-mediated inflammatory diseases, and you recognize readily that the discordance between monogenic twins is profound. 90% of people don't have the same diseases. So, things that spring forward in this area are, obviously epigenetic variation, the effects of the virome, which has this predator-prey relationship,
Yep.
microbiome. How are these interdigitated in the present or future cloud?
Yeah, it's a great question.
Can you summarize the question for the people who may not have heard that on the...
Sure. Yeah, I was going to say it's a complex question. Basically, the question is, there's a lot of additional information, certainly that's well beyond the genome and that's well beyond some of the measures we make, including different aspects of viruses and the immune system and so forth, and how might we think about incorporating those in the future. Shorter. But yes. So I think one is that you point out there's so many dynamic elements here, and that's why so much that's going on here is beyond just the genome, right? And there are some people that really advocate like everything is the genome, and we definitely don't believe that. The second element is you're exactly right. The data clouds that we have, although they are kind of expansive, relevant relative to what has been previously, still leave out a ton of things. And this is one of the reasons why when people push back or they want to, they don't want to make so many measures out of the human body. From my point of view, I think that we absolutely have to, in the sense that our bodies are such complex dynamic systems, as you were alluding to, and the amount of data that we have about them is a pinprick compared to the complexity that's there. I mean, when you drive your car around, it's making 10,000 measures a second about how it's operating and is feeding those back into a computer system, right? And it tells you, your mechanic goes in and it tells you, here's what's wrong. And so we don't even have that on our own bodies, and our bodies are much more complicated than that. In terms of your specifics on immune and viruses, I absolutely think that those need to be incorporated more. I'm a big fan, although we haven't done it yet, of immunosequencing and things like this, where you could have kind of a yearly update of what your immune system's seen and whatnot. I think that over time would be very valuable. And viruses, I I talked a lot with Doug Kell, who used to run the equivalent of the NSF in the UK. And he's very convinced that pathogens are triggers for many different kinds of chronic diseases. And he's accumulated a lot of evidence that would be really hard to see across lots of different papers. Another colleague of mine has got a really interesting one related to a famous disease. Under review at Science, so I won't say what it's about. But there's a number of these that are out there now. And I think the more that we can evaluate that and study that and then figure out what are the most important bits so that you can figure out economic ways to get that in the healthcare system is hugely important.
Yeah.
My interest is in scientific reversal. You're talking about wellness, but we spend 86% of our dollars, $3 trillion, on people who are sick.
Yep.
And the ability to reverse or stop some of these chronic diseases, heart disease, diabetes, what we're doing is using plant-based nutrition, and we're arguing with the actuaries, whether they're self-insured employers. Look what we've done. We've reversed the heart disease. There are no more threats of bypasses and stents in the future. Pay us on an ongoing basis, and we'd like to add your metrics, if we can use these metrics of scientific reversal in that kind of inquiry.
Yeah, absolutely. I think we should definitely talk about that. And I, like you, am also a big proponent of reversal. And early stage reversal, I think, is going to be really key. Even in the Alzheimer's study that we're doing is really focused on early stage reversal of Alzheimer's disease. And Dale's made a big focus on that. And Alzheimer's, I think, is a good case because once you're at severe Alzheimer's and your brain has really decayed, I don't know how you put that back. I mean, I could imagine, some science fiction way in the, with stem cells and stuff in the far future, but immediately, I don't know how you put that back. And so it really does come back to that early stage reversal where you can still do something about it. And I think these kind of monitoring systems for understanding what that early stage reversal, what that early stage disease really looks like, how far back it in fact goes, and then figuring out ways to tap back problems before they really bloom. I think it's going to be a major effort. And reversal is interesting because reversal is a little bit more of a finite problem than prevention. Prevention is very all-encompassing, even though we're very interested in that. But reversal is a little bit more tractable in the short term.
Yeah. Oh, we got to close up. Just to remind everybody, in September 26th, there's going to be another grand rounds with Dr. Suzanne Go, who's pediatric neurologist who's studied autism, looking at the mitochondria and other therapies that can help these children. And also, for those of you who are interested, the work that Dale Bredesen's doing has been documented in his book called The End of Alzheimer, which is coming out August 22nd. And it's really quite a book that details the methodology through which he's reversing Alzheimer's. So thank you, everybody, and we'll see you next time.